We are going to teach you all about RNA transcription, which is the first step in gene expression. Transcription is when genetic information in a molecule of DNA is used as a template to generate a molecule of RNA. This process takes place in the nucleus of a cell. Transcription is broken down into three parts: initiation, elongation, and termination. Initiation is the first step in this process, in which the transcription complex forms. Elongation is the second step in this process, in which nucleotides are continuously added to the new strand of RNA. Termination is the final step of transcription and is when RNA polymerase dissociates and the RNA molecule is released.
The first step of mRNA transcription is known as initiation. There are several steps in this complex process. The first step of initiation consists of general transcription factors binding to the promoter of the DNA strand. A promoter is a region composed of a few hundred base pairs where RNA polymerase and other proteins bind to the DNA strand. Many promoters in eukaryotes consist of a region of DNA known as the Ta-Ta box, since the TA sequence is normally present. The binding of general transcription factors recruit RNA polymerase II, which is the complex responsible for the transcription of coding genes. Next, transcriptional activator proteins bind to enhancers. These transcriptional activator proteins end up recruiting a mediator complex of proteins to the promoter region. After this, the DNA begins to loop, which causes the transcriptional activator proteins to bind to the mediator complex. Now, all the proteins are brought into close proximity with one another, meaning that transcription may proceed.
The second phase of transcription is elongation. By definition, the elongation phase of transcription refers to the process through which nucleotides are added to the growing RNA chain. During this process, RNA polymerase II separates DNA and forms a bubble within the complex. Free ribonucleotides from the surrounding environment are added to the complementary base pair at the 3' end of the RNA transcript. When this process occurs, guanine, cytosine, thymine, and adenine on the DNA strand will pair with cytosine, guanine, adenine, and uracil on the RNA transcript, respectively. This process continues as the RNA polymerase II complex travels along the DNA strand.
The last step of RNA transcription is known as termination. By this time, there may be thousands of nucleotides before they are dissociated from the template. Transcription termination explains how RNA polymerase knows when to stop creating RNA. This process can be defined as when RNA synthesis continues along the DNA template strand until the polymerase encounters a signal that tells it to stop or terminate transcription. The process of termination stops at a sequence known as a terminator, and the transcript is released. After the transcript is released, the RNA must encounter some modifications, known as RNA processing, in order to prepare for translation, the last step of the central dogma. First, the 5' end of the primary transcript is modified by the addition of the 5' cap, which is necessary for translation so that the ribosome is able to recognize the mRNA. Then the primary transcript must encounter the process of polyadenylation, which by definition is the addition of a string of about 250 consecutive A-bearing ribonucleotides to the 3' end, forming a poly-A tail, which helps with transportation and stability. Lastly, certain sequences known as introns are removed from the primary transcript in a process known as RNA splicing, which is catalyzed by a complex of RNA and protein known as the spliceosome. The spliceosome initiates the cutting of one end of the intron and connecting it to a nucleotide near the other, forming a loop and tail called a lariat. Exons are then brought close to each other and the introns are released. This is necessary in order to remove the non-coding regions of the RNA.